Hello, everyone. My name is Deborah Kahn. I'm the founder of Being Patient. For those of you who don't know me, I'm a journalist who has a mom with Alzheimer's disease. When I was working at the Wall Street Journal, my mom was diagnosed with Alzheimer's, and it started the journey for me to discover more information and understand questions like, what have we learned in the last 10 years? And how far away are we from a cure? How is science approaching the search for a cure? Surprisingly, it was very hard for me to find this type of information. So I decided to quit my job at the Journal and launch Being Patient. I called many scientists around the world, from Asia, Europe to the US, to understand more about the complexities of Alzheimer's and what we've learned so far, discovering information like the fact that beta amyloid plaque can be seen in the brain up to a decade or more before one person sees a symptoms of Alzheimer's disease. A lot of this information helped me to understand research, and it was comforting in a way. So that's what led me on this journey, and apropos to my experience and to the topic of today's panel, the pipeline. Today, we are going to have the pipeline to progress, and we have a very distinguished who's who of in drug discovery and development with us today. I'm pleased to introduce Phyllis Farrell. She is the global head of external engagement for Alzheimer's disease at Eli Lilly and Company. Michael Irizarry, who is the Vice President of Clinical Research and the Deputy Chief Clinical Officer of Neurology Business Group at Eisai. Brian Kelly is the President of Payer and Provider Solutions at IQVIA. And Dominic Walsh is the head of Neurodegenerative Diseases Research Unit at Biogen. Welcome, everyone, to our panel, and I'm sure it's going to be a really interesting conversation. As I mentioned earlier, I'm always looking for the answers of where we are in the scientific research. 2021 obviously will not only be remembered for COVID, but for those of us impacted with impacted by Alzheimer's disease, it will also be remembered as the year that the FDA approved the first drug aimed at the pathology of Alzheimer's disease. Although it was a rocky start, Aduhelm was approved, and it has now opened the door to many other later stage clinical trials that are going on, in particular with drugs ending in MAB, which stands for monoclonal antibodies, which target mostly the beta amyloid plaque in the brain. So it was a great change for drug discovery and really opening new doors. But it wasn't the only thing going on in the landscape. We also have a lot of different ways to diagnose now. New biomarkers are being discovered. So I would say 2021 was a good year for movement on the pipeline and taking us closer. But really what I want to hear is from your perspective, from pharma's point of view, what was 2021? Let's look back and say, what is the biggest change that you see in terms of moving forward in development? And Phyllis, why don't we start with you?
Thanks, Deborah. And thanks for being here with us and sharing your personal story. So many of us have been impacted. So I think when I look back at the progress we've made just in the last 5 to 10 years, so Lilly's been at this for over 35 years, but I've only been with the team for about 10. And when I look back, we have made so much progress in understanding the pathology of this disease, understanding how do we study the disease. So typically in pharmaceutical drug development, the trial design is very, very clear, and you're just putting a molecule in to see if it's going to do what you want it to do. In our case, in Alzheimer's disease, we were trying to figure out how to study the drugs. At the same time, we were discovering the drugs. So when we started the Expedition 3 study on solanezumab many years ago, we'd already completed 4 phase 3 studies at that time in Alzheimer's disease. But those 4 studies, as you mentioned, Deborah, they didn't measure amyloid. So we went back and looked later at those studies and realized through some sampling that 30% of the people in those studies didn't actually have amyloid pathology, which means they didn't have Alzheimer's disease. And it probably meant that an anti-amyloid therapy wasn't going to work. So when you think about just that change alone, and now, of course, we've got the advent of the ability to see tau pathology in the brain. And so there's a couple of really big changes that have happened in this last 5 years. And as a field, we're sharing that across all of our pharmaceutical collaborators and research collaborators to make sure we all benefit. The first is that we've probably been studying this drug too late. So just like oncology, you don't want to start therapy at stage 4. You're going to make more impact if you started at stage 0 or stage 1. And in Alzheimer's disease, as you mentioned, we were catching this disease much, much, much too late. So I think that's the first thing. And then I think the second thing is the progress in this pathology in the brain, amyloid and tau pathology. Once again, potentially another oncology analysis or analogy. We're able now to really get at the pathology in the brain and measure that pathology in a real way so that we can minimize the variability that you might otherwise see in cognitive symptoms. These are huge strides forward.
Michael, would you agree with that? I mean, we're hearing so much about the discovery of new biomarkers, or even finding them in the blood. Blood tests are coming out. How important is the role of biomarkers in terms of, identifying patients for trials and actually choosing specific treatments?
Yeah, I would agree with Phyllis that the advances in biomarkers over the past 10 to 20 years has really been remarkable and has helped us better define the pathophysiology of Alzheimer's disease. So that the concept is expanded from a clinical concept of dementia and progressive cognitive and functional impairment to a biological conception of a disease characterized by early amyloid deposition, which then seems to trigger the spread of tau and neurofibrillary tangles and then neurodegeneration. And we have biomarkers now that remarkably can see these pathologies in living people. So Phyllis had mentioned the amyloid PET and the tau PET that can give us a sense of where the amyloid is and where the tangles are in the brain and the timing of when they occur. And now we can see those same pathologies reflected in changes in blood tests. So now amyloid beta protein fragments can be measured in blood tests, phospho-tau fragments can be measured in blood tests, and these correlate with the amount of pathology in the brain. And these are already being incorporated in clinical trials to help select patients, to stage patients, and to identify specific treatments or treatment regimens for patients. Phyllis mentioned that we realized that 20 or 30% of people that we thought clinically had Alzheimer's actually did not have amyloid. And conversely, we've also seen that amyloid begins 15 years or so before the onset of symptoms. So we can now identify cognitively normal individuals that have elevated amyloid and have started clinical trials in trying to treat at that period before there's significant irreversible neurodegeneration. So these are clinical trials in so-called preclinical Alzheimer's disease, where amyloid is identified, elevated amyloid is identified either through PET or even blood tests or CSF, and treatments initiated. And these are long-term studies to understand if this can prevent eventual cognitive decline or progression of Alzheimer's pathology.
Okay, and I should have announced earlier that we have a way for our audience to ask questions. We're getting questions already. Thank you very much for those. So obviously some of you know that you can ask any questions you want during our conversation. So this is a question on being patient. We often get questions from people who really need to know the basics, right? And as science is debating what the right target is, Being Patient recently put together a chart where it's now much more diversified. I mean, the majority is still going after beta amyloid, but we have tau, we have modulating inflammation. We even have people going after microbes to understand whether or not that's a target to prevent the disease from spreading. So this person is actually asking, if you have a very basic question, which comes up a lot, if you have amyloid in your brain, does that mean you 100% have Alzheimer's? And I'd like to also say, the other add to that by saying, if you don't have Alzheimer's in your brain, does that mean you don't have Alzheimer's? So, let's start with the basics. I'm kind of throwing it out. Someone raise your hand and say, I can answer this one, because I know you all have an opinion or can answer it. So the floor is open.
Well, maybe pick up on something that both Phyllis and Michael said, and that is, how we better understand the disease. I think if you initially framed your question in terms of over the last 5 years, this is not an understanding that's been obtained over the last 5 years. This is an understanding that we've developed over decades. And I think one of the big advances recently was actually not so much scientific, but actually a formulation of what we already knew. And that was the 2018 guidelines that the FDA issued, where they actually defined Alzheimer's disease by its pathological hallmarks. Now, that connected two very distant processes. What we see at postmortem in an autopsy brain, and what you can see, which both Michael and Phyllis referred to, by brain imaging using PET imaging agents, where you can look at amyloid and you can look at tau. And to connect those two pieces of information that were so distant for so many years was facilitated by longitudinal studies, that is, natural history studies, where people had collected patient samples over long years. This is efforts like ADNI, like the studies were being conducted at Rush University with the religious order studies, studies like Oscar Hansen in Sweden with the Biofinder study. We now understand the real human disease, not simply by looking at end-stage postmortems, but looking at people in life. And that really helped the FDA formulate the guidelines where we can define Alzheimer's not by obscure clinical terms, but precise biomarker definitions. So we have a 6-point scale going from 1, where a person doesn't have any symptoms, no symptoms whatsoever, but they have the beginning of pathology in their brain, all the way to the end of the scale where you have a person who has filament pathology and has, unfortunately, the ravages of dementia evident in their brain. So I think that, to get at two questions here, go back to the can a person be alive and well with amyloid in their brain and symptom-free? Absolutely, yes. Can a person have dementia and not have amyloid and have Alzheimer's disease? Absolutely no. So it is a requirement for the definition of the disease.
So it's interesting to see that research is really looking to the asymptomatic population for answers, something that we wouldn't have done maybe a decade ago. So tell us, Brian, what are some of the considerations in terms of patients and data that are really imperative to kind of pushing research and that pipeline down to more drug development?
Sure, Deborah. So first, I think you've got, with Phyllis, Dominic, and Michael, you've got incredible scientific domain expertise here. But where I play is really in understanding how to use data to basically help get drugs to patients in a better way. And this is really complicated, and I think our our thinking about this has evolved immensely over the last few years. And Dominic touched on this. I mean, the fact that we now longitudinally look at the natural history of disease and can better define that and understand that has really opened even the FDA and the regulators thinking about the fact that real world data is just critically important to this. If I think about it, you've got a set of data that you're going to need to get a drug approved by regulators, and the pharma companies know exactly kind of these endpoints and the like. But once the drug's been approved by the regulators, it still has to be bought by the patients or the payers, and that's another set of real world data and real world outcomes that are going to have to be demonstrated. Now, the way that I really look at this is that we need to begin to collect more systemically the right data set to meet all of those end goals. And I actually think that some of the work Us Against Alzheimer's is doing to really determine what are the outcomes that matter most to all of the different stakeholders, because you need alignment between what's important to the patient, the caregivers, the physicians, and society in general in terms of what we can afford that need to be aligned. Now, the great news is that I think because of the maturation of technology, we now have tools we just did not have several years ago. We have long longitudinal databases of many different types that we can use to study these important questions and answer them much more quickly. As hard as COVID has been on the world, the one thing I would mention is that COVID has actually accelerated the way that we think about virtual trials and reaching out and the use of other wearables and devices to collect data. So really interesting time to be.
So actually, can we ask you more specifically, what is the data now that is being considered that wasn't maybe 5 years ago? What are we getting? I mean, we're seeing technology zoom ahead. And we're getting a lot of readings if anyone wears a Whoop or Apple Watch. And honestly, I don't think a lot of people really understand some of the biomarkers. But from the scientific side, what data are you getting that is important for drug discovery that we didn't get before or we didn't know about?
So I think we're still early there, and I'd love my colleagues to give their perspectives too. But I think we're still early in the use of what we call, Michael and Phyllis talked about biomarkers, maybe blood-based test or imaging tests that basically say somebody has amyloid or tau or other important biomarkers. People are now beginning to talk about digital biomarkers. Are there data that comes off wearable devices that allow us to infer different information about how that patient is done? I used to be a practicing neurologist before I got really into the data business, and I would see my patients with dementia a couple times a year. If we put a wearable on a person, we have hundreds of thousands of data points that we never had before. Now, what we have to do is we have to do the studies, and they will take a little bit of time, and they've already begun, but we have to be able to clinically validate these different digital biomarkers and say, okay, when I see this digital biomarker, it means this. And I think we're just really beginning that journey right now, and I think that's the opportunity of the next few years.
Okay, and another question has come in that asking, are there unintended consequences of removing amyloid? But I'd like to make that question a little bit bigger since we have such distinguished panelists. As a drug company, how do you decide what the right target is? What types of information validates, developing a drug? I mean, obviously, amyloid is the most popular choice for drug development, but what is going on behind the scenes that validates that? I'm throwing it to all of you.
Let me start,
go ahead.
and then I'm going to let Mike do this one. We'll see if he still agrees with me, because sometimes we do, sometimes we don't. Usually we do. But Deb, I think I want to build on for that question on the comment you made about the analysis that Being Patient did, looking at all the different mechanisms. When you think about that are being studied right now, amyloid, tau, et cetera, when you think about drug development in general, what we have to do is we have to have a really good understanding of the disease state and the disease pathology. Then we find what's called a druggable target. So you identify that target and you say, I can actually interact with that part of the disease cascade in some way with a drug. In this case, obviously, we're talking about monoclonal antibodies as the first ones out of the gate. There was a time where we were fighting between amyloid versus tau. In fact, I think at one point it was called the Baptists versus the Taoists because it was beta amyloid, which was very clever. But what we have found as we've understood these things is these are actually part of the same type of disease cascade, with amyloid coming first, tau coming later, and then the neural inflammation and some of the other things that are downstream from that. The same thing happens in drug discovery and development. If you find amyloid first, you find that it's a druggable target first. You're going to see those treatments come through the pipeline first. But then you see, oh, now tau, it's a new target, and we found a way to make it druggable, and we're starting to see it come through. So I think you'll see it, kind of come in waves as that innovation cycle moves. And I think that's really exciting because what we're going to know about Alzheimer's disease is it's not going to be a one hit. You're not going to have one silver bullet or one mechanism that actually can cure this disease. So moving our way through that disease target, I think, is important. At Lilly, the way we make that decision is to see if when targeting the pathology, do you actually move the clinical endpoint that you're looking to move? In our case, that's cognition. And right now, the only place where we really have that connection, and this is what the accelerated approval was all about, is between amyloid and cognition. So now what we're trying to say is, can you make those connections on these other targets? Mike, how much did I butcher that? Would you edit?
I would agree with that. I expand it conceptually in terms of drug development being seen as what's sometimes, depending on the company and the development process, the 4 R's. So the right hypothesis, the right population, the right dose, and the right endpoint. All leading to pathophysiology guided drug development. So early on, each of those elements need to be considered for each particular target and evidence generated to support that. And the more evidence that's generated to support each of those features, the higher the probability of progressing through development. And then Dominic may be able to talk more about the translation side and the early clinical side for targets.
Yeah, well, so I agree completely with what both Phyllis and Michael have said. So I like the 4 R's in particular, Michael. I think that's a very nice way to kind of keep everything in place with the hypothesis, the population, the dose, and the endpoints. One thing I would add in is, unfortunately, because Alzheimer's disease is such a common disorder, it actually means genetic analysis is very powerful. Because we have very large numbers of individuals, we can actually look across the population base and pick out small genetic factors. And I think that we're getting to a stage where we're actually understanding that, non-familial forms of Alzheimer's disease, so-called sporadic Alzheimer's disease, is actually impacted by multiple different genetic factors. And that gives us a great place to start for what Phyllis referred to as the next wave after A-beta and the next wave after tau. Then we have a whole slew of other targets, the most notable of which is ApoE, but there's certainly a lot of others, and there's a lot of those factors that actually interact with each other. And so I think that we can really start to think, I've been working on Alzheimer's disease since 1991. And when I started, people said, the best you can do is stop Alzheimer's disease. You'll never actually ever be able to treat it. I think that we're getting within sight that we can start talking about really treating people who have symptoms.
Okay, I want to unpack the genetic question. We actually got a question from someone in our audience asking, what is the state of genetic research? Now, just to give our audience a bit of background, APOE4 is the what's known as the Alzheimer's gene, but then we also have discovered early onset markers. There's a few genes that we know if you have that marker, then you're going to have early onset Alzheimer's, APP, SN1 and 2. But we cover on Being Patient all the time new genetic markers that are being discovered. My question is, when we discover a gene makes someone perhaps more predisposed to Alzheimer's, what does that mean in terms of research? Does that mean we're headed to do genetic reengineering, of certain genetics in order to prevent the disease? What does that look like in drug development and research down the pipeline?
Maybe I'll just jump in because I brought up genetics to start off with. I think that we want to be very clear about the different roles that genetics play. There's the so-called autosomal dominant Alzheimer's disease, and that's individuals who will develop Alzheimer's disease because they have causative mutations. Those mutations destine those people to develop disease, right? But there are very small, they're an important but very small percentage of the total number of people with Alzheimer's disease. So there aren't truly, inheritable forms of Alzheimer's disease beyond people who have mutations in APP, which is the precursor protein from which the amyloid beta protein that forms the amyloid in the brain is derived, and presenilin, which is a protein who constitutes one of the enzymes that releases A beta from APP. So if you have mutations in either of those two genes, unfortunately, you're by and large doomed to develop Alzheimer's disease. But the other genetic components don't doom you to develop Alzheimer's disease. They increase your risk. Even people who are ApoE4 positive, who have two alleles of ApoE4, we all have two copies of each gene. And if you have two copies of E4, you're at very increased risk of developing Alzheimer's disease. But even amongst that population, there are some people who do not develop Alzheimer's disease. So that implies that there are other genetic modifiers that can actually turn down the effects of some of these risks. And so understanding the balance between these different genetic modifiers actually holds great promise for the future for people who are even at high risk of developing Alzheimer's disease.
So Deb, just to jump in on that, because I think this genetic question is really, really important. Being able to separate out the two things that Dominic was just talking about, those kind of genetic predispositions, familial, the dominantly inherited Alzheimer's disease versus these risk factors. Because, you also asked a question earlier about if I have amyloid in my brain, does that mean I'm definitely going to get Alzheimer's? Well, these are risk factors. And I think for those of us that have the opportunity to engage externally, it's really important to talk about the things that you can do to potentially modify that risk. Now, as researchers, we want to figure out how to generate that evidence-based data, whether that be through the Alzheimer's Association running the finger study and some of the lifestyle modifications they're measuring, or those of us that might be looking at, say, therapeutic interventions. But I think that's a really important thing because we don't know everything yet about why someone does or doesn't, if they get amyloid, or they do or they don't, if they have these ApoE4 homozygote or heterozygote position. And, while you can never blueberry your way out of Alzheimer's disease, there are things that we can do for our health to help our brains, help our bodies be more resilient. And I think that's a really important thing when we talk in the community.
And if I could add, building also on how genetics are used to support drug development, these genetic risk factors, even though they may increase the risk a very small amount, do support that pathway as a potential pathway for drug development. And in addition to genes that affect the risk of developing disease, genetic studies are now looking at how genes may affect the phenotypic presentation of Alzheimer's disease, how it might affect biomarker levels or biomarker progressions in Alzheimer's disease. So again, helping us understand the various pathways. And even all of these genetic risks can even be pulled together into what's called a polygenic risk score. That can also help assess risk or timing or onset of Alzheimer's disease, or help select patients for clinical trials. So it's a very active area, and genetic support for a target is one of the strongest validation supports for going after that target for therapeutics.
Thank you. Thank you for that, both to all of you. And I guarantee you're going to like this next question, which is, a good segue from what Michael was just saying. Someone has asked, said, my father and both his sisters died of Alzheimer's. I don't have symptoms, but should I get into a trial anyway? So that opens the door to talk about clinical trial participation, how important it is. Diversity included. But let's talk a little bit about people like myself. I too have a mom with Alzheimer's disease. She was diagnosed quite late. Am I a viable candidate now that you all are turning towards the asymptomatic population? Are people like us who have parents with Alzheimer's disease, but so far, knock on wood, don't have any cognitive issues, although my family might tell you differently. But are we candidates? Are we for upcoming research? And how would we be able to contribute to understanding and developing the pipeline?
I think I speak for all of us when I start by saying that clinical trial participants are our heroes. We could not do the work that we do without clinical trial participation. And in fact, when you look at what's the thing that's slowing us down the most these days, it's actually recruiting participants. The science is moving much, much, much faster than the trial operations. And I know you know that because of the questions that you get from the Being Patient group. I can say from our experience on A4, which was the first asymptomatic study that was done in preclinical Alzheimer's disease, still going on at Lilly in partnership with the NIH and several other partners. What we found is the folks that are most motivated to participate are actually those that have a family or friend who's been impacted. So they might be looking at it for themselves, they might be looking at it for their children, or they might be doing it to honor that loved one and just say, I want to be part of the solution. I want to be part of the battle. And because now we've learned so much about this disease, there's actually an opportunity to be part of the solution when you are cognitively unimpaired. There are some of those biomarker tests that you will go through to see whether you would qualify for the study or not, because we would want to make sure that you had that underlying pathology. You still don't want to run a study for 10, 15, 20 years. So you've got to be able to catch people at the right phase of the disease. But there's absolutely the opportunity over a certain age for people to raise their hand and say, I want to be part of the solution. That can be in a drug study, but it can also be in some of these other studies, observational studies, like Brian mentioned, biomarker studies, digital biomarker studies, lifestyle modifications. There's so much opportunity right now to raise your hand and be part of the solution. And word of mouth is often the way participants get referred to a clinical trial.
I just want to add this question that came in because I think it's what people are wondering. So someone has asked, say you're an average 40-year-old, how do you find out if you have one of these biomarkers in order to participate? I mean, I'm assuming it's not left up to the individual to go to the doctor and say, can you check if I have plaque in my brain? So what's your suggestion to the people who don't have symptoms, but maybe a family member? What do they need to know in order to understand whether or not they qualify for a clinical trial?
One of the things we kicked off early on was talking about the tremendous advances that have been made in biomarkers and just the better understanding of the disease. So as well as all of the work that goes on with pharma companies running clinical trials, there's a lot of folks in academia and in clinical centers who are running longitudinal natural history studies. If you don't qualify for a clinical trial, you may consider becoming a part of a natural history study. For instance, there are some natural history studies that are trying to recruit people who are E4, who are completely negative, so that they can follow them over time. And that's a tremendous way both of protecting yourself and giving to the community, because you're giving to our understanding of the disease, while at the same time, if you do transition into that early stage of being amyloid positive, you'll be first to know about it. So I think that that's one thing that we can really, encourage everyone to do. Is there's a lot of very active work out there with natural history studies. I'd encourage people to get involved in those.
So
To be super direct, ct.gov has a listing of studies. It's not incredibly easy to use. So the Alzheimer's Association has their trial match. So you can go and actually put in your characteristics and they'll see if there's a match, as does the Banner Alzheimer's Prevention Registry. Typically, if you're below the age of 50 or 55, you're probably not going to find a therapeutic intervention study. As Dominic mentioned, you're more likely to find one of the observational studies, the natural history study, the behavior or lifestyle intervention. I know that Lilly has a website called Lilly Memory Trials. So you can log on and find a site near you. It's kind of like find a Starbucks near me. You just type in your zip code. It'll show you what's recruiting near you. And I'm sure that ASI and Biogen have very similar tools. But if you don't want to go company by company, there are some other ways to do that. Very, very simple, very direct, very easy. And try it, see if it works, and then spread the word with your friends.
Okay, and I do want to touch on the diversity issue because it's a big one, and everywhere I turn, there's people wanting to get more diverse populations into studies. So, Michael, just tell us quickly, what is it that what would that add to research, having higher representation of Latinos, Blacks in clinical research?
I think it's critically important to understand that the drugs work similarly across diverse populations. It's also very important for us to improve the access to clinical trials. And I think we've learned from COVID that we can do many clinical trial activities virtually or remotely. So home infusions of treatments, remote cognitive assessments, remote safety assessments, and building those capabilities so that we can bring the clinical trials to patients where they are, as well as reducing the burden of clinical trials on patients. So right now, for several of our pivotal trials, the cognitive assessments and visits can be pretty long, and that also limits the access to clinical trials for individuals. So I think the COVID pandemic has accelerated some of these decentralized activities, and then a range of initiatives that involve the community in terms of building trust in communities, bringing clinical trial sites to more diverse communities. So it's a range of activities I think need to be taken in parallel with input from the stakeholders, so input from potential participants.
Brian, I don't know if you can answer this, but is there any data around what the biggest barriers are? Because I mean, I've heard people say of Alzheimer's disease, 85% don't get filled to capacity. Do we have any data around that? Anything that you would offer insight on?
I think it would be hard for me to put a specific number because it's going to be very context specific around a specific study. I think Michael just talked, I think, beautifully about the importance of diversity, and I think to the industry's credit, the first thing you have to do is recognize that diversity is important. And I can tell you, almost every trial that we run has basically targets for different population ethnicities and populations, and we they have to be filled. And so that actually has driven us to really try to figure out, how do we find those patients, how do we engage in those patients, how do we work with those local communities to get those people in. I also agree entirely with what Michael said, is we have to make it easier for people to participate on trials. And the more we can do remotely, the less time you have to go to a clinic or a site for a long assessment. I mean, that's very difficult for people that are working 2 jobs and have a family. So I think we're making a lot of progress there, but there's still more work to be done.
Great.
Yeah,
okay.
There is a white paper that was put out by the University of Southern California, a collaborative event where they actually worked, Brian, with IQVIA and did a really nice funnel that identified what all the barriers were and bottlenecks to clinical trial recruiting. And then they are in the process right now of doing a solution set, and every one of those solutions is they're looking also at diversity. And that becomes really important because when you think about the fact that African Americans are twice as likely to be impacted by Alzheimer's disease and the Latinx population 1.5 times as likely, it's of critical importance that we actually have a represented population in these studies. So if you can anyone can go find that white paper now on the USC website. It's called the Bottlenecks in Alzheimer's Clinical Trials, and has also been published, I think, with Paul Eason as the lead author, and Dana Goldman. And then I'm really looking forward to that next piece of that work, which is going to talk about some of the solutions. Hopefully we'll have some of them already in play, but I think that'll be really helpful too.
Great.
Deborah, I was just going to say, we're getting much better at doing diversity. All of the big COVID vaccine trials had very strict requirements around certain populations, and those all, it took a lot of work, but they all recruited, their targets in incredibly small, short periods of time. So we're getting there.
Okay, and we're almost out of time, but I just really want to kind of wrap it up with looking forward, looking at the pipeline to progress. Now, I think a lot of science has come to agree that Alzheimer's is not just treating one thing, it's treating kind of, and especially with prevention, it's looking at a whole host of either risks or symptoms. I just want to get each of your takes on what is imperative to solving the puzzle in throwing forward in the next 5 years. How do we look at this disease, and how do we really break down some of the barriers to finding how to successfully treat it? Why don't I just start with you, Dominic? I'm going to just do a round robin quickly. Go for it.
Well, you can always accuse me of being over optimistic, because that's the way I like to be. And I actually think that in terms of treating earlier, that we're in a pretty good spot. I wouldn't be surprised that we can have a tremendous benefit to a lot of people simply by targeting A-beta if we go early enough. I think as we progress into the disease, that's where we need to really play upon, multiple different pathways. But I'm kind of like the glass half full sort of guy. And I think our glass is already half full, in that we know that we're starting to see a class effect with targeting A-beta. And I think that if we go earlier, that we'll see even more success.
Great, thanks. Michael?
I think we're seeing a lot of evidence that Alzheimer's as a neurodegenerative disease may be tractable to treatment, and clinical trials reporting out over the coming years will help us understand that. I think what's critical for us to then have an impact is to improve the detection and diagnosis of Alzheimer's disease, whether through biomarkers or digital diagnostics, identifying individuals early in the disease course, or even in the prevention space when they're more likely to respond to potential treatments before there's been very severe irreversible neurodegeneration.
Great, thank you, Michael. And Phyllis, very quickly, I think we're down to a minute.
Just a double exclamation point behind what Michael just said. 30 years ago, we were in a situation where people wouldn't say the C word. And now you can't imagine getting a cancer diagnosis and your doctor not telling you. And yet today, only 30% of people are told when there's an Alzheimer's diagnosis. And that's because the doctors aren't bad doctors, but they're not confident. And now we've got these wonderful tools. I would love 10 years from now for everybody to know a cognitive assessment of their brain health, just like they know their blood pressure and their blood glucose. And for us to be able to say, look, we can actually measure that you've reduced your amyloid pathology the same way we can reduce a tumor. I actually don't think that's 10 years from now. I think that could be next year. We just need to move a little faster in the healthcare systems.
I love that optimistic note. Brian, do you have anything to add?
Yes, I think that in 10 years time, I think we are going to have the digital infrastructure to be able to recruit people virtually to really assess their longitudinal health better. And when we have that, we're going to be able to run trials with much smaller numbers of people that are going to show effect much faster, and that's going to be to everyone's benefit. So I share Dominic's optimism, absolutely.
Okay, and thank you so much. As a daughter of a mom with Alzheimer's, I want to say thank you all for the research and all of the hard work that you're doing. It means a lot to all of us out there. Dominic, Michael, Phyllis, Brian, thank you so much. I found this incredibly helpful and as a check-in, and we look forward to talking to you perhaps next year to see what 2022 brings. Thank you all so much.
Thank you.
Bye.
Bye.